علم القرآن
taught the Quran,
خلق الإنسان
created man,
علمه البيان
and taught him eloquence.
الشمس والقمر بحسبان
The sun and the moon move by precise calculation,
والنجم والشجر يسجدان
and the stars and trees prostrate.
والسماء رفعها ووضع الميزان
And the heaven he raised and imposed the balance.
صدق الله العظيم.
Assalamu alaikum, good morning, good evening, and good afternoon to various participants residing in various continents of this world. I welcome you all on this 2 days online certificate course on basic plant tissue culture techniques, which is jointly organized by Comstech Islamabad in collaboration with International Center for Chemical and Biological Sciences and Pakistan Biotechnology Information Center, Karachi chapter. Now I invite my colleague Khazima for further proceeding. Khazima.
Thank you, Dr. Samad. With this, I request Professor Dr. Iqbal Chaudhry, Coordinator General Comstech and Director ICCBS, to welcome our guest speaker and participants for today's event. Over to you, sir.
Thank you very much. Bismillah ar-Rahman ar-Rahim. Assalamu alaikum and good morning and good afternoon, ladies and gentlemen. With the blessed ayat from Holy Quran, we are starting this very
सर आप सुने मैं आपको काफी चाबी लगा बाद में सर सुने मैं आपको काफी चाबी लगा बाद में सर
Okay, so let me welcome you to this very important event, which is jointly organized by Comstech, which is Ministerial Standing Committee for Science and Technology for the OIC countries, and International Center for Chemical and Biological Sciences, and two of my colleagues, Khazima Muazzam, represent Comstech from Islamabad, and Dr. Samar Yusuf, who is representing ICCBS and also PABIC, Pakistan Biotechnology Information Center, are already here. Now, I've been told that there are participants from 20 countries, which really make this event truly international. And in this difficult time of constrained mobility, such events are extremely important to keep our interest and competency alive in various disciplines of science and technology. This field is very close to my heart. I've been working together with many of my distinguished colleagues all around the world in the promotion of plant biotechnology for the benefit of people. And as we have worked for decades, I think one thing which we have identified is that capacity building is critically important, because you cannot really do anything useful unless people are competent and skilled. Now, there is no doubt in competency. We, as a OIC region, we have lots of young people, over 800 million young people. And there are so much eagerness of learning new technologies. And this is our responsibility then to work together to improve their skills. Plant biotechnology, as we all understand, is one of the most popular, fundamental, and earliest biotechnology, which has led to the foundation of the discipline of plant biotechnology per se. This technology has given the birth of many new technologies. Apart from its own merit of being one of the fundamental techniques of improving crop varieties, rapidly introducing new species and creating disease and other tolerant varieties. Technology itself is extremely important because the skills which this technology has propagated eventually led to other disciplines. This is the oldest one in terms of cell and tissue technology, and this skill has been used in molecular biology. It is used in regenerative medicine, stem cell, whole range of different technologies has emerged out of this skill. So this workshop and skills. I'm sorry, somehow my mic is muted from somewhere, and then I have to unmute it. So I think the emphasis would be on the skill development, because this skill is not only important for you to work in the field of plant biotechnology, but also very important that you can move forward and do research in other fields as well. So this is an extremely important event. I think the most important part of this event is absolutely wonderful resource persons. The one which I really admire, which inspired me through their lifelong contributions, not only the science of plant biotechnology, but also their advocacy, their contribution towards developing skills and promoting the benefit of this very useful technology. This certainly is qualified to be ilm nafe, the knowledge which benefit. I would like to acknowledge my colleague Dr. Samar Yusuf, coordinator of Pakistan Biotechnology Information Center. She has been actively engaged since long in promoting plant biotechnology information in Pakistan and in the region, and that she has worked very closely with my colleague Khazima Mausam. You already see a very nice document, which is the result of hard work of our team. As I said, we have a very competent and distinguished resource person. And with that, I'm sure that you would be able to benefit from this exercise immensely. I would request you to interact as you move forward and make sure that you understand from the science which is going to be presented to you. With that, Sama, you want me to do something else also?
Dr. Saab, but I want you to, that's all. Khazima will introduce our speaker, and then we will proceed for lecture. Yeah.
Thank you very much, and welcome again to this very important event.
Thank you, sir, for your very insightful remarks. With this, we will start our course with lecture number 1, Importance of Tissue Culture Techniques for the Sustainable Development of Muslim World. And this lecture will be delivered by Professor Dr. Behzad Gharyazai. Let me introduce our distinguished speaker to you all. Professor Dr. Behzad is the producer of world's first commercialized insect-resistant transgenic rice and considered as a founder of modern agriculture biotechnology in Iran. In addition to academic achievements and positions such as Dean of College of Agriculture at a state university in Iran, he held executive positions including Deputy Minister and Head of Agriculture Research, Education and Extension Organization of Iran. He has established a world-class Agriculture Biotechnology Research Institute of Iran in 1999 and served as a Director General for about a decade. He is also serving as a member of Higher Council of Science and Technology of Iran, and as well as on the Biosafety National Focal Point. President of Biosafety Society of Iran, and is a member of Public Research and Regulation Initiative Steering Committee. He has been involved in several international negotiations on modern biotechnology and biosafety regulations. With this, I request Professor Dr. Behzad to start his lecture for this course today. Over to you, sir.
Bismillah ar-Rahman ar-Rahim. His Excellency Dr. Muhammad Iqbal Chaudhry, Coordinator General Comstech, Director ICCS, and Distinguished National Professor, Director of PUBIC, my very excellent kind sister Samar Yusuf and Miss Kazima, and everybody attending the meeting. Assalamu alaikum to all of you, and thank you very much for inviting me, and I am very honored by your invitation. Hope someday after the COVID-19 issue is resolved very soon, we will again meet each other physically and embrace each other and say again Assalamu alaikum and shake hand, inshallah. Okay, I am asking your permission to start my sharing my presentation. Please allow me to know if everything is fine or something goes wrong, because we never know if something is going wrong in this type of presentations. The topic of my presentation is the importance of tissue culture techniques for sustainable development in Muslim world, and we will take about 20 minutes to cover the topic and then entertain a few questions if needed. There was a high-level expert forum on how to feed the world in 2050 held in the headquarters of FAO in Rome, and they decided that we really need to feed the 9.1 billion people in very near future, 2050, of which 1.8 million, sorry, this is one digit is wrong, 1.8 billion is today Muslim country. To feed this population, food production must be increased by 70%, and production of safe food is more challenging. Well, His Excellency Ayatollah Khamenei, our supreme leader, joins the others in calling for attention to biotechnology, since it is very important field. Recently, OIC has released a report on agriculture for food and agriculture and food security in OIC member countries. I don't know how can I make my presentation in a way that I can see only my presentation and not the others covering my presentation. Can anybody help me doing that? Because audience is covering my presentation and I cannot really see what I'm saying.
We can see your presentation, brother.
But I cannot see myself.
Yeah, that's what the drawback of online virtual events are.
Okay, let me see. Okay, I was able to manage. Okay. According to this report that is recently released and it is freely available on the net, the OIC strongly emphasizes the development of agriculture sector to attain food security and enhance growth and prosperity. Agriculture remains in many OIC countries as a major source of livelihood and employment. OIC countries are relatively abundant with agricultural resources, such as arable land and human resources, and account for a significant share of global agricultural production and trade. We have also successes. Since 2010, the gross agricultural production index in OIC was much higher than the world average, and that is something to be proud of. Half of the OIC member countries were ranked among the top 20 producers of major agricultural commodities worldwide, and I'm pleased to say that Pakistan is one of them, exporting a lot of good things like cotton, rice, very delicious, lovely mango that I love very much, and tangerine, everything. But we are also facing a lot of challenges in OIC member countries. Agricultural productivity in OIC member countries remains insufficient to feed the growing population. Many OIC member countries heavily rely on food imports to meet their local demand. 10.5% of the OIC total population, meaning 176 million people, are undernourished, or in other words, they sleep while they're hungry. Multiple threats to food security currently exist in many OIC member countries, such as internal conflicts, pest and diseases outbreaks, extreme weather, and last but not least, COVID-19 outbreak. Food self-sufficiency ratio in Persian Gulf countries is not something that we can live with it very easily. As you can see in some of the Islamic countries around us, they are only 0.42% self-sufficient in cereals, which is very bad situation. In total, as of 2014, 2.19% self-sufficiency in cereals was achieved in the Persian Gulf countries. This is a list of maize importing countries that shows, unfortunately, Islamic countries are at the top. Egypt spends $2 billion a year to import only maize. Iran spends around $1.5 to $2 billion to import maize. Algeria, Saudi Arabia, Morocco, Malaysia, Bangladesh, Indonesia, Tunisia, Jordan, Yemen, all of the Islamic countries are importing maize. There is another list of selected countries importing more than $100 million soybean per year. As you can see in here, many of them are Muslim countries: Egypt, Indonesia, Iran, Pakistan, Bangladesh, Malaysia, Saudi Arabia, Tunisia. The Gulf countries, the Persian Gulf countries, currently import up to 90% of their food to meet their rising demand, with the UAE expenditure alone reaching up to $6.7 billion in 2009, according to TradeArabia.com. This is relatively old slide, but I like to share it because the situation is still the same. But to this end, I would like to emphasize that according to Food and Agriculture Organization Millennium Statement, biotechnology can help the situation. Biotechnology provides powerful tools for the sustainable development of agriculture, fisheries, and forestry, as well as the food industry. Biotechnology can be of significant assistance in meeting the needs of an expanding and increasingly urbanized population in the next millennium. There are different fields of biotechnology. When we are referring to biotechnology, we are not really referring to one thing. We are referring to genetic engineering and genome editing that is revolutionizing the agricultural products. For example, Pakistan, the host country, is currently producing about 2.8 million hectares of GM cotton and is exporting it. Microbial biotechnology, systems biotechnology, synthetic biotechnology, molecular physiology, and last but not least, tissue and cell culture. Today, we will focus on tissue and cell culture only according to the title of the workshop and the presentation. What is tissue culture? Because I was told that the audience are blend of experts as well as politicians and students. I'm sorry if something in my talk is very simple for someone or somewhere somehow complicated for the others. But let me see if I can manage. Plants are totipotent. What does it mean? It means that in principle, each and every individual plant cell can regenerate an entire plant. This special feature distinguishes plants from animals. If we lose our hand, we cannot grow it again. But if a plant loses its branch, it can replace it. This is called totipotency. Tissue culture is the art of employing the phenomenon, taking tissue fragments or individual cells from a plant and growing an entire plant on a special media on the bench. Tissue culture, cell culture, meristem culture, anther culture, pollen culture, protoplast culture, all of them are synonymous to plant tissue culture, or they are part of plant tissue culture. Plant tissue culture has many different applications. We can categorize the applications of plant tissue cultures in two major groups. One group is the application of tissue culture per se, like mass propagation of uniform and healthy plants. But some of the other applications of tissue culture are considered as a tool required for other techniques, such as genetic engineering and genome editing. So genetic engineering and genome editing cannot go without the application of plant tissue culture. Therefore, plant tissue culture is a tool also to be used by experts in the field of genetic engineering, synthetic biology, wide hybridization, and protoplast fusion, etc. I will start with mass propagation of plants. This is a very sad situation that you can see in this picture. I am really sad to show that. This is the date palm orchards after bombardment, and this is the end result of war. War does not only kill people, but also destroys their livelihood. So replacement of this date palm is really challenging. Traditionally, basal offshoots are used for date propagation. You can see in here the basal offshoots, the baby plant that comes out of the trunk of the date palm. Disadvantage is that each tree produces very few offshoots, maximum of 12 in its entire life. It is time consuming because offshoots are collected from old trees only, and young trees do not produce offshoots. However, tissue culture can help. You just need to bring in one of those offshoots in the laboratory and take the meristematic section of that one and surface sterilize it, chop it into several pieces, theoretically to millions of pieces, and mass produce plantlets inside the jars, inside the tissue culture media. And then this is where the work is done. This is Rana agro industry. And then you multiply them, you propagate them, and then transfer them to hardening situation. And finally, enjoy the fruits. These are tissue culture derived mature date palms from Hormozgan, Bushehr, Khuzestan, and some other countries. I'm very pleased to let you know that date palm industry is almost entirely controlled by Islamic countries. The world leaders of date palm producers are listed in here, and all are luckily Muslim countries: Egypt, Iran, Saudi Arabia, Iraq, Pakistan, United Arab Emirates, Algeria, Sudan, South Sudan, and Oman. I am pleased to let you know that all of these countries are using tissue culture for mass propagation of date palm to some extent. I have visited the tissue culture companies for date palm propagation in United Arab Emirates, in Egypt, in some other countries, including Iran. Another good example of the application of tissue culture for economic development and for safety and nutrition is the mass propagation of potato. Each and every potato everybody is eating in this world should come from tissue culture first, because potato is affected by some types of viruses that are transferred through insects, and therefore if you grow them again, you will not have enough yield and you will lose your yield. So what we do usually is we take a few pieces of tiny pieces of meristematic region of potato, we sterilize it, we make it virus-free, and then we mass produce them inside the bioreactors. You can see in here, we call them microtuber or mini tuber. Microtubers then are produced under the greenhouses, and produce mini tubers. Mini tubers and are then grown in clean fields, free of any insects, controlled fields, and then they produce different classes of potato seeds. We have used this technology for the self-sufficiency of potato mini tuber. We used to import potato mini tuber, but now we are exporting it. We are producing 12 millions of mini tubers. We have transferred the technology to 22 SMEs, and we have we have been able to save more than $29 million from not importing potato mini tubers and also exporting it. We also export the potato for food, and also by not importing potato, which always comes from with soil and associated pests and diseases, and therefore we prevented the importation of pests and diseases to the country. I am very pleased to show and share with you that Islamic countries are not included or are less included among the major importing potato importing countries. As you can see in here, Belgium, Netherlands, Germany, Spain, Egypt, Malaysia are still importing very little potato. Algeria, United Arab Emirates, and Azerbaijan and other Islamic countries are importing very little. I'm sure that they can become self-sufficient just by using tissue culture for this technology. Mass propagation is used for many different species. I don't have time to explain and go through all of them, but this is very key technology, easy to use. Everybody has the technology, but has massive impact on the economy. Let me introduce some of other applications of tissue culture. Tissue culture is used for in vitro selection. In vitro selection relies on the somoclonal variation. Somoclonal variation is resulted from tissue culture. This is either induced variation, which is a kind of response to some of the chemicals included in the tissue culture media, such as 2,4-D, or they are pre-existing variations that are selected for. For example, in this picture, you can see variegated species that are produced apparently by somoclonal variation and in vitro selection. But we never know if they are genetic or epigenetic unless we go through and test them. Tissue culture is also required for genetic and genome editing. This slide shows two major different methodologies for plant transformation. This is agrobacterium mediated plant transformation, and this one in here is bioelastic bombardment. Both of these techniques require explants that are produced through tissue culture. For example, this is callus. Callus is either produced through immature embryo or mature seed, or even a piece of root or plantlet. And then after transformation, again, we need tissue culture to propagate the single plant that is genetically improved. Another very excellent technology that requires tissue culture is reverse breeding. As you know, all Muslim countries are importing a lot of hybrid seeds. In panel A, you can see that hybrid seed comes from two inbred lines. These are the two inbred lines, for example, in here, Colombia and Landsberg, that are strictly restricted by the companies and nobody can have access to them. They only sell the companies only sell the heterozygote or F1 seed. These heterozygote F1 seeds, if the I'm sorry, something is annoying me. Something is, please move this window away from me. I cannot. Okay. Okay. If crossing over is taking place, you will have this situation. In here, you will never be able to use the same plant to get back to the heterozygote situation, and you will not get good seeds. So you have to go back to the company and buy the heterozygote seeds again. But if you can, in a way, to intervene the crossing over, then you can have millions of several, okay, not millions, many opportunities to regain genotypes that if you cross them, you will again be able to resolve the heterozygote situation, and you can have the reconstructed heterozygote phenotype or genotype and enjoy the seed. That will be exactly the same. Well, you need to go through the paper and explanation. So you can use this technology or other haploid technologies to produce hybrid seeds that are very important for good production. Sometimes in plant breeding technologies, we come across a situation where we don't have proper gene inside the domestic genotypes that is available. So we have to go for other species. We have to look at the proper genes in other species. But crossing two different species from two different genus, or even from one genus, two different species, is somehow difficult. And sometimes you cannot get embryo developed on the mother plant. So in this case, you have to rescue the embryo. There are cases when you cross two species, embryo is developed, like in this case, you can see the embryo. But this embryo cannot survive on the mother plant itself. Therefore, you may take them and grow them on the culture media, and from that you can get a plant that is a cross between two different species. In here, for example, we are showing you with the cut style method of embryo rescue. That is another technique. Synthetic biology also requires tissue culture. Well, I don't know if I will have some time to share with you some clips, but I would rather not. Though there is no report of totipotency for animals, but it doesn't mean that there is no animal or human tissue culture. And in fact, tissue culture is currently used to produce lab-grown chicken meat. This meat that you see in here, and any type of meat you name, can be produced from animal cells in laboratory. And this is not some story or science fiction. This is already available in commercial situation in Singapore. Singapore is the world's first country to approve lab-grown meat for sale, and they are using it. It tastes like chicken, and it doesn't involve slaughtering any animal. Well, saffron is a very, very important spice for Iran. Perhaps, according to the data, we are producing more than 90% of the world saffron. But it is very tedious. From 1 hectare, you can get only a few kilogram or less than a kilogram saffron. So we are thinking of producing this stigma that is the end product of saffron, this one you can see in here, through tissue culture. We have been able to modify different hormones, different chemicals. For example, we have been able to use BA and TDZ and produce this type of heart shape or globular shaped structures, and finally produce saffron inside glass jars or in vitro. Inside glass jars or in vitro. Okay. Another very significant application of tissue culture is to produce medicinal plants inside cells. You see, this is the systematic strategies to increase secondary metabolites produced within plant through cell suspension culture. So what you need to do is to take cells from any of these plants that you can see in here, that they produce some sort of secondary metabolites that are either medicine or they are for